Good morning, the students of class 9. We are dealing with the chapter respiration. In the last class, we have learned about the definition of respiration. We have gone through the chemical equation of respiration. Then we have learned the sequence of steps that occurs during the process of respirations, which are mainly the glycolysis and the Krebs cycle. So in the last class, we also learned about glycolysis and we have also learned about Krebs cycle. Clear? Then we have learned about the anabolic process and the catabolic process, and this particular process we have discussed taking example of respiration and photosynthesis. Then we have discussed the difference between respiration and combustion or burning, and the general surfaces through which the respiration occurs or the parts of the plant through which respiration occurs we have discussed. It generally occurs through the stomata, then it occurs through the lenticels present in the stem and the general surface of the root. So these things we have discussed in the last class. Then we have discussed the types of respiration, which are two types: aerobic respiration and anaerobic respiration, along with their differences. So all these things or topic we have discussed it in our last class. Now today we will learn the experiments on respiration in plants. Clear? So we will try to explain or try to learn the experiments on respiration. So before going into the experiments, the first thing that we should know while performing any experiment or while setting up any apparatus for experiment, we have to learn that we have got two setups before conducting any experiments. Now those two setups are known as experimental setup and control setups. Clear? So before conducting any experiments we have to set up two things. Okay, we must have two setups. One is experimental setup and number two is control setup. Clear? So all the things will be similar in the experimental setup and the control setups. All the conditions will be same, except the thing is that whatever the thing we are going to test, we are trying to prove, that particular thing will be absent in the control setup. Okay, so we can see over here that both are identical, in which every condition is the same except one. The setup in which the condition under study is missing is called the control setup. For example, if we are trying to learn that carbon dioxide is produced during the process of respiration, so we have to prove that carbon dioxide is released by the green plants during respiration. Then in the control setup, clear, in the control setup, those green plants must be absent or it must be placed dead. Clear? So in the control setup, the thing under investigation or under study will be absent, but the thing will be present in the experimental setup. So for performing any experiment made by for photosynthesis or made before respiration, clear, we must have two setup: experimental and control setup, so that our observations can be easier. We can compare our observation, our result between the experimental setup and the control setup. It will be easier for us to observe the results. Clear? When we compare the two setups. So first of all, you should remember this thing: experimental setup and control setup. So let's come back to the topic: experiments on respiration in plants. So the first experiment of the day is experiment to prove that oxygen is used up in respiration. So the aim of the experiment, aim of the experiment is that oxygen is used up by the plant during respiration. Okay, so first experiment, our aim is to prove that oxygen is used up in respiration. So all of us know that while performing an experiment we require certain There are certain requirements. Clear? So in this experiment, what are the requirements? What are the things that we need? So first thing you can see over here, we require two flasks. Okay, flask A and flask B. So first thing what we require is the flask. Clear? Then we require germinating bean seeds and we require the date bean seeds. Clear? So first requirement is that we require flask. We require two flasks, flask A, then we require flask B. Clear? Second requirement is we require germinating bean seeds and require date or boiled seeds. Then we require antiseptic such as carbolic acids. Requirement is to avoid the growth of bacteria. Okay, that we'll discuss when we go through the experiment. So we require antiseptic also such as carbolic acids. Uses we will discuss it afterwards. Clear? Then we require the wet curtain. Okay, then we require soda lime, which is a mixture of sodium hydroxide and calcium oxide. So all these things, all the uses of these things, we will discuss it when we go through the experiment. So these are the requirements which we need. Okay, then also we require a test tube containing soda lime is there only. So first of all, now procedure. Those are the requirements. Class AB, germinating seeds, date bean seeds, antiseptic, example carbolic acids. We require wet curtain to provide water. Then we require a small test tube that contains the soda lime, a mixture of sodium hydroxide and calcium oxides. Now, listen the uses of carbolic acids and soda lime. What are the uses of carbolic acids and soda lime? Carbolic acids are used to avoid the growth of bacteria on the date seeds. One more time. Carbolic acids are used to avoid the bacterial decay, is there only, or to avoid the growth of bacteria on the date seeds. Clear? Because if we do not use this antiseptic, then bacterial growth will be there on the dead seeds. Then those bacteria will respire. They will give out carbon dioxide. Then our experiments will be totally wrong. Okay, we cannot come to our right conclusions. So in order to avoid the growth of bacteria on the dead seeds, we are using carbolic acids. Next, soda lime. Why are we using soda lime? The function of soda lime is to absorb any carbon dioxide. If you look over here, the function of soda lime is to absorb any carbon dioxide released by the seeds. So those soda lime will absorb the carbon dioxide released by the seeds. Now let's go to the procedure so that it will be easier for us to learn their particular functions. Okay, so after requirements we have to go to the procedure. So how we are supposed to start the experiment? So take two flasks. We have to label them flask A and flask B. Clear? In the flask A we will put germinating bean seeds. Clear? We have to put germinating bean seeds. In the flask B we will put date or boiled bean seeds. Clear? So in the flask A we will put germinating bean seeds. In the flask B we are putting date bean seeds. In both the flasks we have added the wet cotton wool. Clear? So that it will provide water to the seeds. But in flask B, along with the wet cotton wool, we are putting some antiseptic. Now, what is the example of antiseptic? Carbolic acids. Now, why are we using carbolic acids in the flask B? So that it will prevent the growth of bacteria on the dead bean seeds. Clear? Now, next thing. In both the flasks, you can observe the presence of test tube. Can you see it over here? We have got the test tube being held in position in both the flask A and B. Now this particular test tube contains soda lime. So in the both the test tube we are putting soda lime. Now what is the function of soda lime? It will absorb the carbon dioxide, all right, present inside the flask. which will be released by the bean seeds. Now you can see the delivery tube also over here. All right, and you can see the level of water in the delivery tube. This is very important. All right, you have to observe the water level in the delivery tube. This is the beaker containing water, and this one is the delivery tube which is being inserted into the respective flask A and B. Okay, now let's come back to the procedure. So after the setup is ready, we will keep it for, let's say, 1 day or 2 day in the normal conditions, in the normal light and the normal temperature. Okay, we will keep it. So after a few days, what will we observe? There will be observations. Requirement is there. Then procedure. We have set up the apparatus, we have set up the control setup and the experimental setup. Now the setup is ready, and we are putting them in a place for a few days in a proper light, in a proper temperature. Now we have to observe it after a few days. That is called observation. Now what will we observe? After a few days, we will observe that, okay, after a few days we will observe that the delivery tube connected with the flask A will show a greater rise in water level. So what will you observe after a few days? After a few days, we will observe that the water level in the delivery tube, that means this delivery tube, okay, the water level in the delivery tube which is connected with the flask A, so the water level in the delivery tube will increase. So this is the particular level of water in the delivery tube. So the level of water in the delivery tube connected to the flask will increase. Now when a burning paper is introduced into each flask, now when we are going to introduce the burning paper in each flask, after removing the cork, this one is the cork, okay, this one is the cork. So when we remove the cork and when we introduce the burning paper in both the flasks, in flask A and in the flask B, the burning paper in the flask A will be immediately put off. Okay, it will not burn in flask A. So as soon as we will introduce the burning paper in the flask A, it will be put off. But in the flask B, it will continue to burn for a short while. Clear? So in the flask B, it will burn for a short period of time, but the burning paper in the flask A will be put off. It will not burn. As soon as we will introduce into the flask A, it will be put off. Clear? So what this will prove? This proves that oxygen was absent in the flask A, thereby showing that it was used up by the germinating seeds. Keep clear, because oxygen helps in combustion. Now since there is no oxygen in the flask A, so the burning paper was not burning. But in the flask B, there was no oxygen because there was no germination, because the seeds that we have used in the flask B are the dead seeds, and you know that the dead seeds does not germinate. So when it will not germinate, the oxygen will not be utilized. So in the presence of oxygen in the flask B, the burning flame will be burned for a short period of time. Clear? But in the flask A, since we have used the germinating seeds, and since the seeds will germinate, it will use the oxygen for germination. And since oxygen are used for germination in the flask A, so in the absence of oxygen, the burning paper will not burn even for a short period of time. So this proves that oxygen is used up in respiration. Clear? And at the same time, we can also observe the rise in the level of water in the delivery tube. Okay, because we are saying that there will be a rise in the water level in the delivery tube which is connected to the flask A, then that of delivery tube connected with the flask B. Clear? This is very important. Now over here the question comes, there are two simple questions which will come under this experiment. First one is that why is there a slight increase in the level of water in the delivery tube of flask B? So you can see over here that there is a slight increase in the level of water in the flask B also, though there is no respiration in flask B, though there is no use of oxygen in case of flask B. But why are we observing the slight increase in the level of water in delivery tube connected to the flask B? The answer will be number one, due to capillary pressure. Okay, we have one answer will be capillary pressure. Due to capillary pressure, there will be increase in a slight increase in the level of water in the delivery tube connected to the flask B. Now, simple things you can like you can just assume. Suppose you have taken a bottle of Coke or a Pepsi. Clear? Let's say you are taking a Coke, Coca-Cola. Then you are inserting a straw into that bottle of Coke. As soon as you enter that straw into the bottle of Coke, you can see the presence of water inside the straw. Bubbles of water will start coming or rising inside the straw. As soon as you dip it inside the Coke, that's simply due to the capillary pressure. So due to capillary pressure, we can see over here the level of water will slightly increase in the delivery tube connected to the flask B. And number two answer can be due to the partial pressure. Okay, because since we are adding soda lime in the test tube, that soda lime will absorb the carbon dioxide. Now, since carbon dioxide is being absorbed by the soda lime, a vacuum will be created. Okay, a vacuum will be created, and that vacuum will generate a pressure which will help in absorbing the water from the delivery tube. So these are the two answers, okay, which will give the solution of the slight increase in the level of water in the delivery tube of the flask B. Number two question, okay, you can see over here, which of the two flasks A and B is a control? So this particular question, I will leave it to you. Just think which one is the control setup, flask A or the flask B. Clear? So briefly one more time, what we're trying to prove? We are trying to prove that oxygen is used in respiration. What are the requirements? Two flask, flask A and flask B. Clear? Then germinating bean seeds and the dead bean seeds, we require antiseptic carbolic acid. Function is to prevent the decay of bacteria or to prevent the growth of bacteria on the dead bean seeds. Otherwise, in the absence of those carbolic acids, there will be growth of bacteria in the dead seeds, and that bacteria will respire. It will give out carbon dioxide, which will hamper the result or observation of our experiment. So that's why we are using carbolic acid. Next thing, wet cotton wool, so that it can provide water in both the flasks A and B. Next thing, we are using a test tube which contains the soda lime. Function is to absorb the carbon dioxides. Clear? Procedure. We are taking two flasks A and B. In flask A, we are putting the germinating bean seeds. In the flask B, we are putting the dead bean seeds. Then in both the flasks, we are putting the wet cotton wool to provide water. But in flask B, we are also putting the carbolic acids. You know the functions. Clear? Then both the flasks are connected with the delivery tube. Leave the apparatus for a few days in a normal condition, normal light, normal temperature. After few days, we'll observe that the level of water in the delivery tube in the flask A will increase up to a certain level. But in the flask B, only a slight increase will be there. And when will we introduce the burning paper into the flask A and B by removing the cork, which has covered the flask. In the flask A, that burning paper will be put off. Clear? It will not burn. As soon as we will introduce into the flask A, it will be put off. But in flask B, it will continue to burn for a short period of time. Now this will prove that in the flask A there was no oxygen. Oxygen was used up by the plant, by the seed for the process of respiration. But in flask B, since there are dead seeds, there was no respiration, oxygen was present over there. And the questions was there why there was a slight increase in the water level in the delivery tube of flask B due to capillary pressure and due to the vacuum that is created by the presence of soda lime, which will absorb the carbon dioxides. And number two, out of the two, which one is control setup, A or B? This will be a question that answer it. Which will be the control setup? Setup number A or setup number B? Just think of it and answer. Clear? Now, next one, we'll try to prove that carbon dioxide is produced during respiration in germinating seeds. So what is the aim of the experiment? Our aim is to prove that carbon dioxide is produced during respiration in germinating seeds. Okay, so we are trying to prove that carbon dioxide is being produced by the germinating seed during respiration. Our first experiment was to prove that oxygen is used up in respiration. Clear? Our second experiment is that carbon dioxide is produced. It's not used, but it is produced during respiration by the germinating seeds. So what are the requirements? You can see over here two flasks, flask A and flask B. Okay, first of all, think of the requirements. What we require for the experiment? Two flasks, flask A and B. Then wet cotton wool so that it can provide water to the seeds. We require the soaked seeds such as pea seed or bean seeds in flask A. Okay. Then at the same time we also require the dead or boiled seeds in the flask B. So requirements simple. Till here, what are we using? We require two flasks A and B. We require germinating seeds. We require dead seeds. We require the wet cotton wool. Now, since we are using the dead seeds or the boiled seeds, so in order to prevent the growth of bacteria, what we require, what we need, we also need an antiseptic. And what is the example of antiseptic? Carbolic acids. Function: to prevent the bacterial growth on the dead seeds. Clear? Because you know that if bacteria are present, then it will show respiration and carbon dioxide will be produced, which will hamper the experimental results. So in order to prevent the growth of bacteria on the dead seeds, we are using carbolic acid. Clear? Okay, now let's come back to the requirement section. Okay, or let's go to now in the procedures. So take two flasks A and B. You can see over here flasks A and B. In flask A, put the germinating bean seeds. In the flask B, put the boiled or dead bean seeds. Then we have to put the wet cotton wool in both the setup. Clear? Okay. Now the mouth of both the flasks are covered by the help of a cork, C-O-R-K, cork. Okay, so the flasks are securely corked and left in similar conditions in light and temperature. Clear? Okay, so in flask B we are also putting antiseptic, that is carbolic acids. Clear? Okay, now both the setups will be kept for a few days in normal temperature and light. Now after a few days, what will be our observation? What will we observe? After a few days, we'll observe that the seeds in the flask A will be found to have clearly germinated. Obviously, it was living seeds, so the seed in the flask A will germinate. Clear? But the seeds which are in flask B will show no sign of germination. Why? It was boiled seed or dead seeds. So the seeds in the flask A will germinate, but the seeds in the flask B will not germinate. Now number two, the gases in each flask are then tested by removing the cork and tilting the flask over a test tube containing lime water. It is very important. Now we have to test the presence of gas in each flask. Now how can we test the presence of gas in each flask? First of all, we have to remove the cork. Clear? We have to remove the cork, and after removing the cork we have to slightly tilt the flask over the test tube containing lime water. So you can see in the picture over here. So after removing the cork, we have to slightly tilt the flask in a test tube containing lime water. So this is the test tube, you can see over here. This is the test tube, and the test tube contains lime water. So we are slightly tilting the flask in the test tube containing lime water. Why? Because we are going to test the presence of gas present in the respective flask. Now the expected gas, carbon dioxide, being heavier than the air, would flow down into the test tube. So when we will shake the test tube, since carbon dioxide is heavier, carbon dioxide is the gas we are going to test. Now since it is heavier than the air, it will settle down below the air. Okay, and remember that the test tube contains lime water. Okay, so the gas from the flask A will turn the lime water milky. That means when we will introduce the gas from the flask A into the test tube containing lime water, the color of lime water will change. It will turn into milky. Okay, the color of lime water or the lime water will turn milky, showing the presence of carbon dioxide. While when we will introduce the gas present in the flask B into the test tube, there will be no change or there will be no effect on lime water. So this proves that germinating seeds will give out carbon dioxide due to the process of respiration. Simple. Okay, so briefly. So we are trying to prove that carbon dioxide is produced during respiration by the germinating seeds. That is our aim. We have to prove that. Requirement was two flasks A and B. We require germinating bean seeds and the dead bean seeds. We require wet cotton wool. We require antiseptic carbolic acids. Then we require a test tube containing lime water. Clear? Now, why are we using lime water? Lime water, when it comes in contact with the carbon dioxide, it will turn into milky. Okay, so it will help us to identify whether there is carbon dioxide or not. Okay, so let's go back to the procedure now. So we have taken the two flasks A and B. In the flask A, we have taken the living seeds. Let's say we have taken the well soaked seeds, which will germinate. Then in the flask B, we have taken the dead seed or the boiled seeds. In both the flask, we have taken the wet cotton wool so that it can provide the water to the seeds. But in flask B, we are using the antiseptic carbolic acid in order to prevent the growth of bacteria so that it will not hamper our experimental procedure, our result. Okay, so both the setups will be tightly covered by the help of a cork, CORK cork, and we'll leave this setup in a normal conditions, in a normal light and normal temperatures for a few days. Clear? After a few days, what will be observed? Our observations will be that the seeds in the flax A will germinate, but in the flax B will not germinate. You know the reason why? Because seed which was used in flax A was well soaked seed, but the seeds which are used in the flax B was dead seeds. Clear? Next thing, we have to prove the carbon dioxide is present in both the flask. Now, what we have to do, we have to remove the cork. Okay, so first we'll remove the cork from the flask A. Then we will slightly tilt it and we'll put it into the test tube which contains lime water and we will shake it. Now, since the expected gas under testing is heavier than the normal air, it will settle down. So when we are putting the gas from the flask A into the test tube that contains lime water, the lime water turns milky. Now, why does lime water turns milky? Because it has come in contact with the carbon dioxide. So when the lime water turns milky, it proves that carbon dioxide is released by the germinating seeds due to the process of respiration. But when we are opening the cork of flask B and when we are putting it into the test tube containing lime water, there will be no change or there will be no effect on lime water. We saw that there is no carbon dioxide. So this experiment proved that carbon dioxide is being released or is being produced by the germinating seed during respiration. Now there is an alternative method for the same experiment. Okay, what we record over here, you can see over here, we have got conical flask over here. Okay, we have got two hole rubber stopper over here. So you can see the pipe being connected over here. Okay, so now, so you have to focus on this diagram. Clear? So I'll just explain the procedure. So just look in the figure very carefully. Flask A, all right, flask A contains potassium hydroxide solution. KOH means potassium hydroxide solution, so that it can absorb the carbon dioxide, okay, present in the air. So you can see the air being introduced into the flask. Can you see over here? The air is being introduced into the flask A. Now since flask A contain carbon dioxide, so all the carbon dioxide which are present in the air will be absorbed. Okay, so that it will ensure that the air which is going into the flask B, that's very carefully, so that it will ensure that the air which is going into the flask B will be free of carbon dioxide. Clear? Flask B will be free of carbon dioxide, and flask B contains lime water. Okay, flask B contains lime water, and if the air which was passing from the A to B contains carbon dioxide, then the lime water will change into milky color. It turns milky. But since we are using KOH, potassium hydroxide solution in flask A, which will absorb all the carbon dioxide, okay, so that the air which is going from the A to B will be free of carbon dioxide, and since it is free from carbon dioxide, it will not turn lime water into milky. Clear? So now there's simply lime water which is not milky. Now the air free of carbon dioxide from B is moving to flask C. As you can see that the flask C contains germinating seeds. Okay, so now the air free of carbon dioxide is moving from the flask B to flask C. So there is germination, there is respiration. So when the air from the flask C moves to the flask D, now the lime water present in the flask D will turn milky. Now why does the lime water in the flask D turns milky? Because the air which was moving from the flask C to flask D contain which gas? Carbon dioxide gas, which was being released by the germinating seeds during respiration, and that particular gas, carbon dioxide, will turn lime water into milky. And this proves that carbon dioxide is released by the germinating seeds due to the process of respiration. So this is one of the alternative method to prove that carbon dioxide is produced during respiration in germinating seeds. Clear? So remember over here we have taken the four conical flasks A, B, C, D. A contains KOH, potassium hydroxide solution, which will absorb all the carbon dioxide. Clear? So when we are introducing the air into the flask A, the carbon dioxide present in that air will be absorbed by KOH solution, and the air free of carbon dioxide will go to flask B, where it will not turn lime water milky because there is no carbon dioxide. And when that air is moving from B to C, clear, it moves from B to C, C contains germinating seeds. Clear? Now from C the air is being transferred or is moving into conical flask D, which contains lime water. So as soon as the air from C moves to D, the lime water present in the conical flask D turns milky. Why? Because in flask C we have got the germinating seeds which releases carbon dioxide during respiration, and that carbon dioxide will turn the lime water milky, indicating that, proving that carbon dioxide is released by the germinating seeds due to the process of respiration. So our aim of the experiment has been proved by these two methods and by these two experiments. Hope it's clear for you. Okay, next one, number third experiment. We'll just discuss about the three experiment. Okay, next we'll do in next class. Third experiment is to prove, or our aim is to prove, that carbon dioxide is produced by the green plants during respiration. Now how to prove it? Okay, simple. Apparatus. What are the things that we require? We require a well potted plant. You can see over here we have got the well potted plant over here. Okay, so example you can use is a geranium. You have to take a well potted plant. Okay, then this potted plant will be covered by the help of bell jar. The entire apparatus has to be placed on a glass sheets. Okay, and all the rims should be airtight so that the air cannot enter the experimental setup. So it has to be airtight. All the rims should be airtight, and it's done by the help of Vaseline. We have to use the Vaseline to make it airtight. Okay, this is carefully. So we have to make it airtight by the use of Vaseline. Okay, now procedure. The outside air is drawn into the apparatus with the help of an air pump. So you can see over here. So from this direction, we are using the air. So air is being drawn into the apparatus, okay, by the help of air pump. So air is being introduced into the experimental setup. So you can see over here we have got two flasks, A and flask B. Now over here you can see the presence of soda lime, okay, the place from where we are introducing the air into the flask A. Clear? So this particular air, it will travel through or it will passes through the soda lime. Okay, so the air which is being introduced from this side will pass through the soda lime, and this soda lime will absorb the carbon dioxide. Function of soda lime, okay, soda lime will absorb any carbon dioxide present in the incoming air. So whatever amount of carbon dioxide is present in this air will be absorbed by the help of soda lime, so that the incoming air into the flask A will be free of carbon dioxide. Now flask A will contain lime water. It contains lime water, and in the absence of carbon dioxide, it does not turn milky. So you can see the lime water clear. Why? Because the incoming air which contains carbon dioxide oxide needs to pass through soda lime. So soda lime will absorb all the carbon dioxide present in the incoming air. Clear? So in the absence of carbon dioxide in the incoming air, the the lime water present in the flask A will not turn milky, it will be clear. Clear? Okay. Now now that air from the flask A will move to the bale jar that contains a well potted plant. You can see over here carbon dioxide free air. Okay, so that air from the flask A will move to the bale jar containing well potted plant. So as the air leaves the bale jar and passes through another sample of lime water, that means when the air from the bale jar, it passes through the bale jar into the another flask B that contains lime water. Listen carefully. Now the air which is present in the bale jar is moving from bale jar to the flask B. Now since the air which is moving from inside the bale jar into the conical flask B, at this particular time, the lime water present in the conical flask B turns milky. Why? Because the plants gives out carbon dioxide during respiration, and the air which is being given out by this plant moves into the conical flask B. The lime water present in the conical flask B will turn milky, indicating that carbon dioxide is present in the air. And from where did the air came? The air was released by the green plants due to the process of respiration. And due to the presence of carbon dioxide in the air, that carbon dioxide will turn the lime water milky. But certain precautions need to be taken while conducting this experiment. First thing is that this experiment needs to be carried out in the dark, or the bale jar, this particular site, okay, or the bale jar which contains the plant needs to be covered by a piece of black cloth to prevent photosynthesis. Clear? So briefly what we have done, we are trying to prove that carbon dioxide is being produced by the green plant during respiration. So we require two conical flasks A and B. Okay, we require a bale jar, we require a well potted plant. Okay, so now what we are doing, so we have taken a well potted plant, example geranium, which is covered by the bale jar. Entire setup is kept on a glass sheets. Now in the conical flask A, clear, which contains lime water, we are introducing the air by the help of the air pump. Now that air which is being introduced into the conical flask A needs to pass through the soda lime. The function of soda lime is to absorb all the carbon dioxide present in the air, so that air will be free of carbon dioxide. So when the air will move to the conical flask A, then lime water will not turn milky, it will be clear because there is no carbon dioxide in air. So there is free carbon dioxide. Alright. Now that air will move into the bell jar that contains plant geranium. Okay. Now again the air from the bell jar that contains the green plants will move into the another flask B which contains lime water. Now as soon as the air from the bell jar that contains the green plants moved into the conical flask B, lime water present in the conical flask B turns milky, indicating that the green plants will give out carbon dioxide during the process of respiration. And what are the precautions needs to be taken before conducting this experiment? Number one, this experiment will be carried out in the dark, or if we are not able to carry out this experiment in the dark, then the entire bell jar that contains the green plant should be covered by a black cloth in order to prevent the process of photosynthesis. Clear? Because carbon dioxide is one of the raw material for photosynthesis. Clear? So today we have discussed about three experiments. Our first experiment was to prove that oxygen is used up in respiration. Second experiment was to prove that carbon dioxide is produced during respiration in germinating seeds. Over here we have gone through the two methods, and last one we have learned about that carbon dioxide is produced by the green plant during respiration. Now next experiment we will discuss it in the next class. So this was for today. Thank you.